Okay, what's up everyone? This is the first online lesson for Bio 100. I'm gonna give this a shot. Let me apologize, my neighbor is doing something, hammering something, and I don't know what's going on, but he's making a bunch of noise, so if you hear a bunch of hammering in the background, that's him. So anyway, we were talking about meiosis, which is the formation of gametes, eggs and sperm. This is the cell division that creates gametes. Okay, and remember, we've got basically 2 rounds of division. Unlike mitosis, which is only 1 round of division, right? And so we went through in class, so we have meiosis 1, which includes prophase 1, oops, not meat, meta phase 1, and then anaphase and telophase 1. Telophase 1. Okay. And then the cells actually divide into 2 in the first round during cytokinesis. So this creates the first 2 cells, and then those cells we're going to divide again in meiosis 2. Okay. One of the important things to remember is that during prophase 1, the homologous chromosomes line up together. Okay. And I had someone ask me after class the other day that they didn't really understand the idea of a homologous chromosome. So, and it's pretty important for you to remember that, or to understand that. Otherwise, this is going to probably not make a lot of sense. So let's go over that again. Homologous chromosomes, they have the same genes, but they can be different forms of those genes. Maybe different versions of those genes, let's say. Okay, so you get one from your one copy. We've got 23 times 2, so we've got 46 chromosomes in total. 23 of them you get from your mom, and 23 of them you get from your dad, right? So let's just take one, and so these 23, each one has got a homologous chromosome. It's kind of like its non-identical twin, right? So let's just take one chromosome that you get from your mom, and then the homologous chromosome that you get from your dad. The one you get from your mom, let's say that this is the gene for, say, eye color. Maybe I shouldn't have picked orange, because orange isn't really an eye color. Let's say that you get the gene from your mom that says that you should have green eyes, and the gene from your dad says that you're going to have brown eyes. So they're essentially the same gene on different chromosomes, but one version would encode for green eyes and one is going to encode for brown eyes. Okay. We talked about the whole earlobe thing. So let's say your mom's chromosome has the gene for the earlobes being attached, and your dad chromosome has the gene for your earlobes being not attached. Okay? So, again, same genes. So this is going to be the ear lobe attachment or not gene here, and then the eye color gene here. Okay? And again, you've got, this is just one chromosome. You've got 22 others, or I guess 21 plus your sex chromosome, 21 others that have different genes for how many fingers you have, or how tall you're going to be, or how hairy you're going to be, or whatever those genes might be. One way that I've taught this is using the analogy of silverware. So this is a spoon, right? That's also a spoon, but it's a different type of spoon. Okay? So they do the same thing, but they're different versions of the same thing. Here's another one. Here's a fork. Here's another fork. They're homologous, right? So they both do the same job. They have the same function, but they're just different versions of that utensil. Okay? So I don't know if that helps or not. So let's go back. So during prophase, the homologous chromosomes, so that let me draw this real quick. In mitosis, the homologous chromosomes, I'm just going to draw them the same color. The homologous chromosomes don't line up together. In meiosis, they do. So they pair up with each other rather than separately. And the purple ones, okay? So that's a big difference between mitosis and meiosis. And remember, I'll continue this later. When they line up, they undergo what's called crossing over, where the homologous chromosomes exchange segments of their DNA. With each other. Okay, and we'll talk more about this later on, but this is why it's important for when the homologous chromosomes line up, like they do here, those two purple ones are going to cross over and exchange little bits of DNA so that the genes kind of jumble up a little bit. Okay. This lining up is called synapsis. Okay. Okay. So let's go over meiosis to the second part of meiosis. Okay. Same stages. We're going to have prophase 2 metaphase 2, anaphase 2, and then finally telophase 2. During prophase 2, the spindle, this is the contraption that latches onto the chromosomes and starts moving them around. The spindle forms and moves the chromosomes towards the middle of the cell. Of the cell. Okay. During metaphase 2, so this is prophase 2, metaphase 2, the chromosomes align themselves, or are aligned, I should say, along the midline, just like they do in metaphase 1 and metaphase in mitosis. Now, this is another kind of different and important step. So now we have the homologous chromosomes have been moved into separate cells. So now we're just left with the sister chromatids, right? So the sister chromatids now separate. And they move to opposite sides of the cell. Opposite sides of the cell. And then, just like in mitosis, during telophase, the nuclear envelope starts to form around the chromosomes, the chromatids. And now we have formed 4 haploid cells are formed. Okay. So here's kind of an overview. We start with one cell. This is meiosis 1. The first bit. The homologous chromosomes pair up. And I'm just going to draw 2. So this is prophase 1. Then we go to metaphase 1, and they line up along the midline. Let me actually draw this, because these are actually they're tetrads. If you remember, they've duplicated, so now we have 4 copies of the chromosomes. Anaphase 1. So this is where the homologous chromosomes separate, but there's still 2 copies there, right? Then during telophase and cytokinesis, telophase 1, the 2 cells are made have the sister chromatids, the duplicated DNA. Okay, now each one of those cells in meiosis 2, I'm just gonna, so we start with 2 cells, I'm just gonna use one of these, right? And then we go to metaphase 2. And this is going to be anaphase. Let's just finish this. Anaphase 2. And then telophase 2. So here, the chromatids, the sister chromatids, line up in the middle. And now in anaphase, those 2 are going to separate. So they're going to be 1 copy of a chromatid on each side of the cell. Then, when they split, they've only got 1 set of the chromosomes, so these are haploid. Okay, and because we've actually got 2 here, we actually are going to end up making 4 haploid cells. Haploid gametes. So those are gametes. Okay. So let's go back and just compare mitosis to meiosis real quick. So in mitosis, again, the homologous chromosomes do not line up together. So that's one point. And you create 2 identical daughter cells. They're genetically identical. In meiosis, we've got a couple things. So the homologous chromosomes do line up, as they're showing here. Together. And in this one, we're creating 4 similar but not identical. And these are haploid gametes. Similar but not identical. And then these are diploid cells. That's another important difference. 2 identical diploid, I spelled cells wrong. 2 identical diploid cells in mitosis. And in meiosis, we're creating 4 similar but not identical haploid gametes. Okay, it's a big distinction between those 2. Okay, I'm going to stop there, and then this will probably end up being like at least 2, maybe even a 3-segment lecture. So you're not like totally, you can take a break. So I'll catch you in part 2.